Right, sir.
Hello, good morning, boys. And today we are going to have demo for experiment number 2 for class 11 biology students. And experiment number 2 is stomatal distribution. Now we need to perform this to understand the structure of stomata. I am sure being enthusiastic boys, you want to see actually how stomata will be live, you want to see. Normally we can't see this, though they are present on both the upper surface as well as lower surface of the leaf. As all of you know, this is the upper surface of the leaf, this is the lower surface of the leaf. So stomata will be present on both the sides depending upon the type of plant. Like you have got monocots as well as dicots. So I will not say in which one it is. It will be there on the upper surface, which one will be there in the lower surface. So you need to do it and tell me in the calculation or result. Right? So as part of it, as usual, the headings are same. First subheading is A. Okay. To study the distribution of stomata on the upper and lower surfaces of leaves and calculate stomatal index. This is the aim. Why are we doing this experiment? So there has to be an aim. Aim is this. And then to perform that experiment, we require certain materials. What are the materials required? Of course, without compound microscope, we can't do anything. So we need to observe at a compound microscope. So we need compound microscope. We need fresh leaves. So you can collect fresh leaves. Then petri dish, ink pillar, then you got slide. You can read this slide, and of course cover slip. Then there's a dye which we put. There's coloring substance, saffron, and then we can also have glycerin. Okay, so that just to avoid air bubbles. Last time I told you, and of course brush, forceps, needle, and to cut these surfaces we require blade. So these are the materials that require. That is fresh leaves, compound microscope, blotting paper, petri dish, beakers, needles, brush, ink pillar, slides, cover slips, blade, pair of tongs, saffron, glycerin, and water. So these are the materials that we require. And procedure, what you do, that is, already I gave a caution to you that since you are performing the experiment, procedure you should write in the past tense and point wise. Don't write stories, don't write paragraphs, right? In past tense and point wise. That is, like first point, a fresh leaf is taken. Now, this leaf has to be fresh and it is devoid of any disease, any fungal disease. So, fresh green leaf is taken. After having taken this, what you require? You need to just tear this like this. When you tear this, you can see a thin plastic type layer here. So that layer, you keep it on the slide. This is slide. On the surface of the slide, you keep it. With the help of blade, okay, cut that transparent membrane in this way. Cut that. And then with the help of needle, don't damage the tissue, slightly you separate it. Okay? And after separating it, now what you need to do, with the help of brush, you put it in the center of the slide. With the help of the brush, hydrate the tissue. Do not dehydrate. This is one of the precautions that you need to take. So put water, 2 to 3 drops of water you put, so that the tissue will be hydrated. After that, with the help of brush only, take this saffron solution which we have put it in the petri dish already. Put 1 or 2 drops of saffron solution like this. And then wait for at least 5 to 10 minutes. So that, what is the purpose? Why should you wait? The tissues like epidermal cells, guard cells, stomata will absorb that dye and we can see the slide clearly. directly. So when this is going on, what is that we need to recapitulate? The theory part of it. That is, what is stomata? What are the parts of stomata? And what are the functions of stomata? Is stomata having a specific thing? And a bit higher is there any ion or radical that decides opening and closing the stomata? Is there any purpose in opening and closing the stomata? So these are the things that we can ponder over when these cells are absorbing the coloring substance. So ponder over that, and all the questions that I posed, answers are there, except I know that I have not answered which is the matter, element or ion that influences opening and closing of stomata. So this is thinking level, hearts we say, higher order thinking skills. Let me see who will answer that. Now meanwhile what I do, I take the cover slip, okay, and put 1 or 2 drops of glycerin so that air bubbles will not be there. The purpose of adding glycerin is to avoid air bubbles. Now put this cover slip, and I told you practicals means 2 things very essential. One is skill, another is patience. You must have a lot of patience. So with the help of 2 needles or with 1 needle and brush, reverse the brush. This is the brush, reverse it, and give the support of this brush to the edge of the cover slip, and slowly you remove the needle as slowly as possible. Slowly you remove. Yes, and ensure that no air bubble will be there under the cover slip. What will happen if there is air bubble? If there is air bubble, when you place it under the microscope, everything will be like footballs and basketballs. That is not our purpose. Our purpose is to see stomata, open stomata, closed stomata, and to calculate how many are there, how many stomata are there. Based on the number of stomata, we will calculate the stomata index, both on the upper surface as well as lower surface. Now, place this slide under the microscope as I have already placed. Now, when you are placing it, ensure that before you place the slide, there is mirror. Okay, adjust the mirror. Keep on seeing through the eyepiece. This is eyepiece. Adjust the mirror in such a way that you will see broad white background in that. That means sufficient light is being allowed, and that gets reflected. Now place the microscope in the center of the stage. Now again, I told you always focus under low power. So 10x, 10 into 10. That means we are making every cell of the leaf 100 times bigger. That is low power. So place it under low power and just keep on moving the slide with the cover slip. At one place you will see certain object moving. Now you rotate the rough adjustment first. This bigger wheels are there, no? That is rough adjustment. Then after you are satisfied, then rotate the finer adjustment or smoother adjustment. You will see further clarity. You will have further clarity. Now rotate that. Once you are seeing, take at least 2 to 3 minutes. View all the cells that are appearing. The stomata, epidermal cells you will see. What is that you need to see under observation? First to see this guard cells. The guard cells are there in the stomata? Yes. What is the shape of the guard cells? Beans seed shape. Cashew nut, cashew nut shape. And some will be dumbbell shape also. Based on that we differentiate which type of plant it is. So see whether the cells the one that I am seeing is the beans seed shape. And then if stomata is open, you will see the stomatal aperture or pore being open. In some I am seeing the stomata has been closed. That means some are open, some are closed. Then also see chloroplast within the guard cells, and then of course nucleus you will see. Then epithelial cells with different shapes, normally cylindrical shape, spongy shape cells will be there. All these you need to observe. So we come under this observations. And after writing all your observations point wise, now you need to put a tabular form. What is the tabular form? You make 1, 2, 3, 4, 5 columns. Serial number, surface of the leaf, is it upper surface or lower surface? So you write first upper, second lower. Number of stomata, how many? Number of internal cells, how many? And stomatal index. So the stomatal index you can calculate with the formula number of stomata divided by stomatal index SI is equal to number of stomata divided by number of stomata plus number of epidermal cells into 100. That gives us stomatal index. Okay? So you need to calculate number of stomata. You need to add number of stomata and number of epidermal cells here and multiply that with 100 to get stomatal index. So this is what you need to do under observation. What is your result? Again, in the result, if you are able to see both stomata on both upper surface as well as lower surface, then it is monopod leaf. Now I am telling that answer. Now if you see stomata more number on the lower surface as I am saying, then it is dipod leaf. So based on the number of stomata where they are present, we can know whether it is monopod leaf or dipod leaf. Now we come to precautions. What are the precautions that we need to take? Number one, I said leaf should be fresh leaf, healthy leaf, green leaf we should take. Number one. Second, when we are adding, I said when we are adding dye, okay, when we are adding the dye and adding glycerin, putting the cover slip, ensure that no air bubbles will be there. What happens if air bubbles are there? I said you will see basketballs and footballs instead of epidermal cells. So ensure that no air bubbles will be there. And third, if there is excess, with experience of course you will add required quantity. But if in case you are adding excess water, excess stain, excess glycerin, then you can use blotting papers. With the blotting paper you can remove the liquids. And then section, you are taking the teeth, ensure that it will not damage the tissue, the very tissue, with the blade or with the needle. So again, that's what I said, you require skill. So carefully separate it. And most important one is, when you place it on under the microscope, particularly high power, the gap between the slide and the object lens will be so narrow. I am demonstrating that to you. You can see here. Yes. Virtually, you can see the gap very narrow. So if you operate the rough adjustment, there are chances of slide getting broken. Hence, under high power, you always need to operate with smooth adjustment, not rough adjustment. This is another precaution. So this is how we need to take. Whenever we are performing an experiment, we need to take precautions. Theoretically, what is that we need? After the precautions, you need to draw, sketch and draw. Now I told you, whatever you are writing, you write on the pages with the lines, ruled page. Whatever tables are there, like observation is in tabular form. The tabular form should be on the white page. Diagram should be on the white page. And draw most beautiful diagrams, colorful diagrams, and label it. While labeling also I told you, try to label it on one side. Don't put 360 degrees here. And then also I told you, when you are labeling it, arrow mark should be towards the part. Arrow mark should be towards the part, not reverse. So that when you are writing, when I am reading see chloroplast, I am just saying the arrow mark is indicating that this is chloroplast. So arrow mark towards the part. So if you present this in your record, that will be good. And then the theoretical part, of course, I told you already, transpiration, water gets evaporated through stomata and that becomes the moisture in the atmosphere. Second, exchange of gases. That is, carbon dioxide goes inside, oxygen comes out, and that oxygen we are breathing. So thanks to stomata. Without stomata, maybe we would have got the amount of oxygen. So basically, transpiration takes place in three forms, as all of you know. One is the cuticular transpiration. The transparent membrane, cuticle, which is present on the surface of the leaf. And that will be around 1 to 2% of transpiration. And some it ranges up to 10%. And then the second one is there are specialized cells called lenticels. So these lenticels also will help in throwing out the excess water from the leaf. So we say lenticular transpiration. And the most important one is stomatal transpiration, which is to the tune of 80 to 90%. Hence, that is the importance of stomata. In our next experiment, next week, we will go with the another one. Until then, keep drawing the diagrams, try to understand the concepts and understand the link between the theory that we have done